Hi, my name is Clarissa, and I'm a junior at NMSU majoring in biochemistry. I'm part of the Schuster Lab over in the biology department, and I'm going to talk briefly about what the Schuster Lab does and about the project I will be working on. The Schuster Lab studies how the microtubule and actomyosin cytoskeletons are regulated during cell division and cell development. My project focuses on cytokinesis, the last stage of cell division where the two new daughter cells are split apart. Cytokinesis is an interesting process because it involves both microtubules and actomyosin. The actomyosin contractile ring drives the cleavage process in cytokinesis. The assembly of the actomyosin contractile ring depends on the organization of microtubules. If the microtubules are disorganized, the factors that regulate the contractile ring assembly shouldn't function properly. However, the cells are still able to compensate. The central spindle is a key player in cytokinesis. It's important in telling the cells where to contract. Many proteins are required for assembling the central spindle. One of these proteins is called PRC1, which stands for Protein Regulator Cytokinesis 1. PRC1 is a microtubule crosslinker. This means that during anaphase, PRC1 organizes the microtubules in an overlapping and anti-parallel manner. The central spindle is formed and cytokinesis occurs properly. Here's what happens when PRC1 is depleted. The cells look like they are dividing, but they leave behind something that looks like a tail, which is called an intercellular bridge. Even though PRC1 was depleted, the cells are still able to complete a good amount of cytokinesis. This begs the question: is PRC1 not essential for cytokinesis, or are the cells performing traction-mediated cytokinesis? Now, what do we mean by traction-mediated cytokinesis? So the cells are living on very stiff plastic, which means that the cells can grip against it. If we take a second look, we notice that the cells seem like they're crawling away from each other. It seems like they're using traction to divide. So if these cells that are able to complete a good amount of cytokinesis are put on a softer matrix, then what would happen? We hypothesized that when cells have a compromised central spindle, the cells use traction to divide. To test our hypothesis, I'm going to deplete cells of different central spindle factors, then observe how these cells divide on a gradient of substrates that range from very stiff to very soft. Since stiff substrates allow cells to generate traction and complete a good amount of cytokinesis, we predict that soft substrates will cause cells to fail earlier in cytokinesis. In summary, we're examining how important traction forces are in cytokinesis when cells have a compromised central spindle.